All right, today we're going to dive into one of the absolute cornerstones of neuroscience, electrophysiology. Now, what is that? At its heart, it's the study of the electrical properties of cells. But for us neuroscientists, it's something much more profound. It's how we listen in on the brain. It's the method we use to decode the actual language of neurons, those tiny electrical sparks they use to think, feel, and act. So, take a look at this. This is a modern electrophysiology rig. And yeah, it looks pretty complicated, but its job is actually beautifully simple. It's built to solve one of the biggest challenges in all of science. How do you listen to a conversation between 86 billion neurons, all talking at once, using tiny millisecond-long electrical pulses? This machine lets us do just that. It lets us tap into that conversation in real time. So here's our game plan. First, we'll cover the basic physics behind the brain's electrical code. Then, we'll look at a neuron in its two main states, when it's quiet and when it's firing a signal. After that, we'll get into the fun stuff, the actual tools we use to record these signals. And finally, we'll talk about how we make sense of all that data and what it actually tells us about the brain. To understand the language, you first have to understand the speaker. And in the brain, the speaker is the neuron. Its entire physical structure, its very shape, is designed for one thing: sending and receiving electrical messages.
Okay, just a quick refresher on what we're looking at. A neuron gets its inputs over here on its dendrites. These inputs cause tiny little voltage changes. Now, if enough of those little signals add up and reach a critical point right here, at the start of the axon, the neuron decides to fire. It sends an all or nothing electrical blast, the action potential, all the way down its axon to connect with the next cell. So there's a really clear flow of information. But what's a neuron doing when it's not firing a message? Is it just off? Well, not at all. It's actually holding very still, maintaining an electrical charge in a state we call the resting potential. And this baseline state is absolutely crucial for everything that comes next. Think of a resting neuron as a tiny, charged-up battery. It's got potential energy ready to go. And how does it build that charge? Well, it's a constant balancing act. You've got this little machine, the sodium-potassium pump, working all the time to push positive sodium ions out and pull a smaller number of positive potassium ions in. This creates a situation where there's way more potassium inside the cell than outside. Now, the cell membrane also has these leak channels that are just for potassium. So, driven by that concentration difference, potassium starts to leak out of the cell, taking its positive charge with it. This leaves the inside of the cell slightly negative. This continues until the negative charge inside is strong enough to pull the positive potassium back in, perfectly balancing the chemical push outwards. That balancing point is the resting potential, right around negative 70 millivolts. So the formal definition is the electrical potential difference across the membrane when the cell isn't excited. But the key thing to remember is that this isn't a passive state. The neuron is constantly burning energy to maintain this charge. It's the stored energy that gives the neuron the power to fire a signal later on. And this table really shows you what we're talking about. Just look at those differences. There's about 10 times more sodium outside the cell, and over 30 times more potassium inside. Each one of these gradients is like a dam holding back water. All that stored potential energy is just waiting for the floodgates, the ion channels, to open up, which will turn that potential energy into the kinetic energy of an electrical signal. All right, now for the main event. If the resting potential is the silence between words, the action potential is the word itself. It's the fundamental all-or-nothing electrical pulse that neurons use to shout messages reliably across the long distances of the brain. Let's break down this iconic waveform. We start at rest, down at negative 70. And input comes in and pushes the voltage up to a threshold, around negative 55 millivolts. That's the trigger point. At that moment, bam, a whole bunch of voltage-gated sodium channels fly open. Sodium ions rush into the cell, making the inside suddenly positive. That's the super steep rising phase. Then, at the very peak, two things happen. The sodium channels slam shut, and a different set of slower potassium channels open up. Now potassium rushes out of the cell, and the voltage plummets back down. In fact, it overshoots a bit because those potassium channels are slow to close. That dip is called the refractory period. It's like the neuron has to catch its breath before it can fire again. So, let's just quickly recap that sequence. We start at rest, and input pushes us to threshold. That triggers the rapid rise, followed by the fall. And finally, there's that little undershoot, the refractory period, before we're back at rest and ready for the next signal. Okay, understanding the theory is one thing, but how in the world do we actually watch this happen in a living cell?
Well, that brings us to the really cool part, the tools and techniques neuroscientists have developed to eavesdrop on these incredibly fast and faint electrical signals. Broadly speaking, you can listen in on neurons in two ways, from the inside or from the outside. Intracellular recording is like sticking a tiny microphone right inside a single neuron. You get a super clean, detailed recording of everything that one cell is doing. Extracellular recording, on the other hand, is like placing a microphone in a crowded room. You're not listening to one person, but to the combined chatter of a whole group of nearby neurons. It's less invasive, and it's great for understanding how groups of cells work together. And you can see that difference really clearly here on the left. With the intracellular technique, the electrode literally pokes through the cell membrane. On the right, for extracellular, the electrode just sits nearby in the fluid between the cells. What it's picking up isn't the action potential itself, but the shadow of it, the flow of ions that creates a tiny brief voltage dip in the space around the firing neuron. One of the most powerful ways to listen from the inside is a technique called patch clamping. You use a microscopic glass pipette to form an unbelievably tight connection with the cell membrane. It's called a gigaseal because the electrical resistance is in the gigaohm range. This seal is so perfect, it electrically isolates a tiny piece, or patch, of the membrane, letting us get ridiculously clear, high-fidelity recordings from just one neuron. Now, when you're patch clamping, you can run the experiment in two main ways, voltage clamp or current clamp, and it's really important to know the difference. In voltage clamp, we, the experimenters, control the voltage, and then we measure the current that flows. This basically lets us ask the question, okay, at this specific voltage, which ion channels open up? In current clamp, it's the other way around. We control the current we inject, and then we measure how the neuron's voltage responds. This lets us ask, how does this neuron react to this specific input? So here's a classic voltage clamp recording. You can see on top, we command the voltage to jump from -70 up to 0 millivolts. The bottom trace is what we measure. See that quick downward dip followed by a slower upward curve? That first dip is the inward rush of positive sodium ions through their channels. It's an inward current, which by convention we plot as a negative value. Now let's look at the flip side, a current clamp experiment. On the bottom, you can see we inject a little pulse of positive current. And what happens? The top trace shows the neuron's natural response. The voltage drifts up until it hits threshold, and then the cell's own machinery takes over and fires a perfect textbook action potential. This shows us exactly how the neuron turns an input into an output spike. So we have these amazing tools. We run our experiments, and we get a bunch of squiggly lines. Now comes the really critical part. How do we decode this data? How do we turn these raw electrical traces into actual, meaningful information about the brain? A raw extracellular signal like this is actually a mix of different conversations, but with some clever filtering, we can pull them apart. If we use a low-pass filter, we get these slow rolling waves. That's the local field potential, or LFP. It's like the general murmur of the crowd, the combined synaptic activity in the area. If we use a high-pass filter, we isolate these sharp, fast peaks. These are the individual shouts, the action potentials from neurons right next to our electrode. But even after we've isolated the spikes, we're still hearing from multiple neurons at once. To figure out which spike came from which neuron, we have to use a computational process called spike sorting. It's basically a set of smart algorithms designed to untangle all those overlapping voices. And this is what that looks like. The whole idea is that because of a neuron's unique shape and its exact distance from the electrode, its action potential has a unique fingerprint. The spike sorting software finds all the spikes and then groups them together based on their shape. Each one of these colored clusters you see here represents the complete train of spikes from a single individual neuron that we've successfully isolated from the crowd. So we've gone all the way from the basic physics of a neuron to these sophisticated ways of recording and decoding its signals. But why do we go to all this trouble? What are the big questions we can finally start to answer? Honestly, the applications are massive. We can connect a neuron's genetic makeup directly to its electrical behavior. We can map out brain circuits by stimulating one cell and seeing which of its neighbors responds. We can try to crack the neural code. How do patterns of spikes represent the things we see and hear? And, really importantly, we can look at diseases like epilepsy and see exactly how their electrical signaling goes haywire, and then test whether a new drug can fix it. To wrap up, electrophysiology gives us this amazing high-speed lens to look at neural function from a single molecule all the way up to brainwaves. It lets us directly test our ideas about how the brain computes. Now, it's not perfect, it can be invasive, and we can only listen to a tiny fraction of the brain at once. But the future is all about combining these techniques with other amazing tools so we can listen to and even talk back to entire brain circuits. And that really just leaves us with one final profound question.